Hello, I'm Hye-jin Kim from Tomocube. Today, I will introduce you to recent studies about analyzing holotomographic images using our new software TomoAnalysis and deep learning-based cell segmentation model for automatic cell segmentation of three-dimensional cell images. When the light passes through the transparent material, phase change is dependent on the refractive index of the material and medium, as well as thickness of the sample. After the plane wave passes through the sample, the shape and the state of the object transmitted through the changes are reconstructed. Because the cells are transparent and most of them are made up of protein, we can acquire images and measure the physical values based on the refractive index. So I think the QPI technique can be a solution for long-term live cell monitoring and analyzing. We can acquire live cell images without any fluorescence labels on the culture dishes using HT. You can acquire living cell images in 3 dimensions for a long time. You can recognize the cells and subcellular organelles by refractive index distribution in these videos. The refractive index distribution lacks molecular and organelle specificity. When you use HT to take images of cells, contrast occurs due to the difference in refractive index, as you can see now. So you can see the cell shape and morphology. You can get a 3D cell image by mapping the refractive index through rendering as well as 2D images. From HT imaging, the cytosol and nuclear membrane and nucleolus are easily distinguished by differences in refractive index. The bright white color in cytoplasm is mostly lipid droplets with a refractive index of 1.37. To expand this idea, we've been trying to construct a robust AI model for the automated and simultaneous segmentation in subcellular structures in holotomography with higher accuracy and precision. We also wanted to try to design a prediction model without molecular marker-based ground truth. This is the detail of the workflow. The subcellular segmentation model predicts 4 different features: cell boundaries, nucleoli, and lipid droplets. Cell segmentation model generated the cell instance mask, cell boundary mask, and 3 subcellular organelles from unlabeled live cells. The cell segmentation model performed well in adhesion cell lines, which were not used for training. What you see now is the result of cell segmentation performed by the AI model in the real software TomoAnalysis. These are the captured results of the cell segmentation for A549 cells from the original HT images. The software can show the segmented masks as you can see. The model also performed well to suspended cells like human lymphoblast K562 cells. This is the page screen of the mask editor in TomoAnalysis, which researchers can edit their masks as they want. Researchers can edit the system-generated masks using various convenient tools. The AI model can directly apply to the numerous biological applications. First, we chose LPS-induced macrophagy activation as a test image because it shows very dynamic morphological changes from a suspended light to an adherent. We confirmed that the volume and surface area of whole cells and lipid droplets increased gradually during activation process for 8 hours. Measurements using these masks confirmed that the surface area and volume of cells and lipid droplets increased over time in Figure B. This is an actual screenshot of the result of a batch analysis performed in the TomoAnalysis. After doing analysis, the report app shows the representing graph of individual cells and a group, and data table including all parameters. We can export the result graphs from TomoAnalysis, and we can also see trends of the experiments and track the outliers by clicking individual data points. We confirmed that the model performed well in the various cell types like primary cells, mouse astrocytes, and human monocytes. TomoAnalysis, a new analysis software optimized for analysis of HT data. The enhanced AI cell segmentation model is integrated in the TomoAnalysis. TomoAnalysis can segment the single cell from 3D cell images and also distinguish its subcellular organelles, nucleus, nucleolus, lipid drumlet using morphological characteristics and range of the refractive index. And it can make subcellular organelle masks. We can do the single cell measurement from 3D masks. The calculating parameters are volume and surface area, projected area, dry mass, and mean RI. Since the image data basically has a huge number of data, I think it should be easy and fast to review the images at once and classify it according to the experimental conditions. To satisfy these conditions, we provide a thumbnail view that allows you to review a large number of data at once, and you can compare multiple data sets simultaneously. In order to batch analysis, it is designed to facilitate the process of grouping data by experimental conditions. Usually, researchers acquire one control group and more than one experimental group. The TomoAnalysis made this conditional grouping possible and configured to perform batch analysis at once. The batch analysis result is a significant amount of numerical results. But using TomoAnalysis, you can check the tendency of each group at once on a graph. Graph can be a very useful tool for checking experimental results immediately after analysis. Lastly, I would like to emphasize that TomoAnalysis is not a software that performs a specific function, but a software platform that analyzes HT images. Therefore, you can continue to add new automation analysis modules independently of existing applications. TomoAnalysis is a software platform that can continue to grow if many researchers use it and give a lot of ideas for the application they want. Thank you for your attention, and we hope to get a lot of interest from you.